It's Anna here from NHCSR. It is wintertime and we are planning our field season. I have come across an intriguing idea and I really do need some input to figure out how to approach this. So in my mind, in my experience working with commercial vegetable growers, tomato is a much more challenging crop from a pest management point of view than pepper. I'm thinking bell pepper versus tomato. Tomato has more serious pest problems than pepper. Pepper is pretty easy. So what gives? What is this? This might be a matter of perception to me, right? So maybe more people are growing more tomatoes. It's a higher value crop. It represents a bigger return on investments for a lot of small scale direct to consumer farmers. Maybe I'm hearing more about tomato because it's more valuable to more people than pepper. But there might be something else going on here that impacts biocontrol in these two different crops. We all know this, but it bears repeating. Biological control is managing pests by letting some other biological organism do the dirty work for us. So this is predators that eat these pests. This is parasitoids that lay their eggs inside or live inside the pests that we want to control, or it could be diseases of these pest species. Oftentimes, these biocontrols, these natural enemies, these agents, they're just out in the field living their life, and it's our job just to let them do that work for us. We call this conservation biocontrol. This can be an issue if you are maybe isolating plants from their natural enemies because you're growing inside a greenhouse, inside a high tunnel. You'll see some problems where you're unintentionally excluding the natural enemies of the pests. This could be if you're using broad spectrum insecticides to control those pests, they can have non-target effects on the natural enemies. So there's a matter of maintaining a balance there. And then there are many options for deploying a kind of mercenary army. We call this an inundative approach to biocontrol because we can release an unnaturally large amount of these natural enemies into that crop to control an unnaturally large outbreak of the pest insects. However, there is a known conflict that exists in tomatoes specifically between the bottom-up effects of plant defense and the top-down effects of biocontrol strategies. I found this paper that does a good job of summarizing this. So anybody who has worked with a tomato plant or many tomato plants knows that they are sticky. You get black. They're chemically very well defended, and all of these chemicals that are on the surface of plants are coming from something called trichomes. So tomato leaves are famously covered in trichomes, which are basically like just hairs. There's different kinds of trichomes, however, and here's a nice picture. So there is a physical impediment to very small predators that are running around on these tomato leaves. Like think about running around in a forest versus in an open plain. So if you're a very small predator, this could be an impact. But on top of those hairs, there's also other chemicals being exuded into that environment. Acyl sugars is an example. Sugars, they're sticky. There's a ton of chemicals, methyl ketones, sesquiterpenes that are all associated with pest resistance. Those can complicate life for very small predators too. The most commonly used species for control of the mites that attack the plant and for thrips are phytoceid mites. We'll see tons of these animals for purchase, very easy to buy them. You can have little sachets, put them out in your plants, and those mites will come out of the sachets and come out to attack. But they've been shown to be negatively impacted by these tomato trichomes. So there are some recommendations for using this in bedding plants. When you start using them in tomato plants, the release rates for those are much higher because of this. So you can see why the differences between tomato and pepper, where pepper, it doesn't have as complicated of an environment on its leaf surface than tomato. There's some interesting things going on here. What can we measure here in our field trials? Well, first of all, we could measure what predators we are observing in tomato and pepper in a field condition. So that conservation approach What's already out there? This review is discussing all the major players of greenhouse tomato in the Balkan region. Out in Europe, there's going to be a completely different complex of beneficial insects as well as pest insects, but it did highlight 2 species of tomato-adapted predators that I was unfamiliar with. These are 2 mirid species, and they are considered one of the most important biocontrol agents for pest control in greenhouse tomatoes in that neck of the woods. And honestly, I have spent many an hour going through tomato samples, trying to record who was present on these leaves, and I definitely saw mirids. I made a note of it. I know mirids can be important, but I didn't really have my eye set on for it. So we could be taking a census of all these insects that we find on tomato, find on pepper, just to see what the species are, to see who's out there. We could take it a step further and collect some of our most commonly observed species on these 2 crop types and confirm that they are predators of key pests of these crops. Something I really like to do is these little bioassay arenas, these deli cups that have a hole in the bottom so we can pop a tomato leaf or a piece of plant tissue that has a flower on it. We can pop that into a water wick that hangs out below, and then you have this arena where you can cage all of your animals together and observe what happens. There's a possibility. Is that interesting or important to anybody to confirm who's out there eating these pests? Let me know. Where else do I need input? I want to know what pests are the most interesting and important to you. Thrips are definitely a hard to control pest. Not everybody has a problem with thrips, but the people who do have problems with thrips have a very bad problem with thrips. They are really famous for being resistant to insecticides. So biocontrol is a really nice approach to controlling a hard to control pest. But again, that's not a big issue for everybody. There's certainly a debate about how serious thrips injury is. In low populations, you'll see this gold flecking. It can definitely make fruit look kind of crappy, but how big of a problem is this for you? If not thrips, maybe spider mites. So spider mite outbreaks, they can cause very serious problems in tomato and pepper, particularly when conditions get hot and dry. So if you're growing in an environment that's hot and dry, you may consistently see spider mite outbreaks. So you might be familiar with this webbing. Once the populations get high enough to produce webbing, it's too late to do anything from a biocontrol point of view. But thinking back, you see that over and over and over again. It might be time to start scouting for spider mites and considering biocontrol. Let me know if this is something that's in your wheelhouse. If not spider mites, maybe caterpillars. We talk a lot about hornworms doing a number on plants. There's certainly a ton of other caterpillars that can do indirect damage on leaves. And there's a ton of caterpillars that do direct damage and bore right into the fruit. Are you seeing this? Are caterpillars an issue for you? The first thing that comes to mind, trichogramma wasps. They're something that you can buy and deploy for caterpillars. I know that there are some negative effects on foraging for these tiny little wasps in tomato. Is that interesting? Another way to approach this is to ask what biocontrol agents are the most interesting to you. Here's a nice summary from Bilbo and Walgenbach about how to use Persimilis mites to manage 2-spotted spider mite. This species of predatory mite is native to Chile, but it was accidentally introduced into Germany in 1959. And since then, it's been distributed globally as an important biocontrol agent in greenhouses for all the vegetables we're talking about, as well as in field-grown strawberry. This is a really important and kind of easy to use biocontrol. According to this summary, Persimilis is recommended to be released shortly after the detection of 2-spotted spider mite when populations are below 0.5 mites per leaflet, and they recommend a release rate of approximately 20,000 per acre. That sounds like a lot. You would never see this number of mites occurring naturally, but you can buy them and release them, no problem. Nonetheless, this paper does indicate that you can get by with a lot fewer if you're on top of your scouting and conduct releases in areas where you're detecting these initial outbreaks, something like 2,500 Persimilis and in 0.14 acre sections of the field, and they'll naturally disperse. That is one recommendation. Are you interested in whether or not that's appropriate for pepper? I don't know. Is this an interesting question? Maybe you're more interested in rates for the generalist predators that are recommended for spider mites and thrips. Is that something that you use? Would you like to know if you're using the right release rates, if you're using them for tomato or for pepper? Are you more interested in rates for the specialist parasitoids that you might be releasing for whitefly, or the specialist parasitoids that you might be releasing for aphids? Please let me know. Any comments or questions are warmly welcomed, and thank you so much for listening.